A-Level Biology 9700, Paper 4, Variant 1, May-June 2025. Second video on this paper, and in this which we will discuss the question number 3. And question number 3 is, porphyria is a group of rare genetic disease in which molecules called porphyrins accumulate in the body. 3 examples of porphyria are X-linked, variegate, and congenital erythropoietic porphyria. So X-linked protoporphyria is caused by a mutant allele located on the X chromosome. So figure 3.1 shows the pattern of inheritance of X-linked protoporphyria in one family. So the family has been shown. You can see the affected mother, which is on 1, and 2 is the father without disease. And they have 4 children. And out of 4 children, the children number 3 and 5 both are affected with the disease. 4 and 6 are without disease. Number 3 is a female. Number 5 is a male. So X-linked porphyria is described as sex-linked disease. They have concluded. Now we have to conclude. Use the information in figure 3.1 to make one other conclusion about the pattern of inheritance of X-linked protoporphyria. Give evidence to support your conclusion. We have to support our conclusion. Say that what is the pattern of inheritance in this. And you can see over here, we have to conclude. First of all, we have to conclude. Our conclusion is that it shows the X-linked dominant X-linked dominant inheritance. It is dominant because it is on X chromosomes. How we can say that it is on X chromosomes? And you can see the mother who is at 1 is affected with disease. And number 3 is daughter also with the disease. Number 5 is son with the disease. It means mother has, you can say, is with disease. So sons inherit their X chromosomes from their mothers only. You know, XX pattern of inheritance, you can say. The female has both XX and male has both X and Y. So if mother XX, female, if from father side Y, then there will be a son. So in this case, it means the X chromosome, which this son who is at 5 number has got the X from the mother side. Because the fathers only transfer the Y chromosome to the son. So X chromosome has been transferred from the mother side. So it is X-linked dominant inheritance. Y chromosome, shows the. So if there should be an X-linked recessive disease, so infected female would normally require 2 mutant X chromosomes, 1 from each parent. So in this way, I would like to say this is the X-linked dominant inheritance. I would like write that sons inherit their X chromosomes from mother. So a mutant X can be passed to sons and cause disease. Whereas father is unaffected and only able to transfer Y chromosome to his sons. So simply, we can say that the pedigree shows X-linked dominant inheritance because an affected mother transmits the trait to son and there is no father to son transmission. An X-linked recessive explanation is unlikely since the affected female would require an affected father. So here in this case, move towards the next question. Next question is variegate porphyria occurs in 1 in 100,000 people in Europe. In the 1600 century, approximately 100 Dutch people migrated from Europe to North Africa. 100 Dutch people. So the population of people of Dutch descent in South Asia, Africa is now 2.5 million. So from 100 Dutch people, it is enlarged 2.5 million. Variegate porphyria occurs in 1 in 1,000 people in this population, suggest and explained why variegate porphyria is more common among people of Dutch descent in South Africa than among people in Europe. So we are moving to, first of all, I would like to discuss what is the uh, uh, what happens over here. According to me, as it shows the pattern of founder effect. So first of all, I would like to say the increased frequency of variegated porphyria is, if we compare it with the Africa and South Africa, Europe, is a classic example of the founder effect. And founder effect is a type of genetic drift. So first of all, I would like to discuss what is founder effect. So founder effect is a type of genetic drift that occurs when a small group of individuals break away from a larger population to establish a new one. As we know, the whole, instead of whole population, only a small group of population is transferred or it goes somewhere else. So this new population may have significantly less genetic diversity than the original population because if the number of population is less, it means they have less variability, they have less genetic diversity than the original population. So the founders are a random sample of the original gene pool and may not have had a representative mix of all the original alleles. So maybe they have those alleles, they have transferred those alleles which are you can say are not good alleles or they have less diversity. So in this way, they can produce more people with the disease as their number has been increased over there. So by keeping in mind this founder effect, I would like to, so the first point, which I would like to say that is the founder effect. So the Dutch settlers who migrated in the formed a small founding population. So first point I would like to write over here is the founder effect. This is the completely the founder effect. Small number of these founders carried the rare recessive allele for the disease, variegated porphyria. And second, genetically they has been isolated. So second point can be the genetically this is a genetic isolation isolation which prevented gene flow. Which prevented gene flow from the larger European gene pool. This allowed the effect of the initial high frequency to be maintained and compounded over time. So this allowed the effects of initial high frequency to be maintained and compounded over time. High privilege in South Africa is not due to any selective advantage of the allele, but to random chance that that a founder carrying the rare allele was part of the very small group that established the new population. So that can be the reason, and which can be due to their transfer from one place to the other, as they had migrated from one place to the other. Now the next question is, microarrays can be used to detect various forms of porphyria caused by mutant alleles. And describe how microarrays can be used to detect a disease by analyzing gene expression. So the question is about the microarrays, and I would like to say that these microarrays are used to detect a disease by analyzing gene expression based on the question, as you can see in the uh, part C of this question. So this technique allows scientists to compare the activity of thousands of genes simultaneously between a healthy cell and a diseased cell. So how do these microarrays can analyze the gene expressions? So the microarrays are also known as gene chips, work by comparing the level of messenger RNA production, which is a measure of gene expression in two different cell samples. So for this purpose, first of all, they isolate the messenger RNA. The messenger RNA in extracted from both the control cells, or it can be represents the genes that are actively being transcribed and expressed. So with the help of this, they synthesize the complementary DNA. The complementary DNA using the enzyme reverse transcriptase. So this makes the molecules more stable for analysis. And beside this, the complementary DNA from the control sample is labeled with one color of fluorescent dye is used, and the complementary DNA from the test sample is labeled with a different color. So in this way, the microarray is a microarray chip contains thousands of microscopic spots, each containing millions of copies of a specific single-stranded DNA probe. So in this way, the labeled complementary R samples are mixed and applied to the microarray chips. The labeled complementary DNA sequence hybridized to their complementary probe sequence. So the third point can be the washing. uh, the chip is washed to remove any complementary DNA that did not hybridize. So in this way, the color of the fluorescent at each spot indicates the relative level of gene expression. So again, I'm going to these some points. Uh, how microarrays can be used to detect a disease by analyzing the gene expression. So first of all, they are gene which comparing they work by comparing the level of messenger RNA production, which is a so first of all, the first point which we can write that is microarrays work by comparing looking very difficult to write right now, making so many other so work by comparing the level of level of messenger RNA production. Which is measure of gene expression. In two different samples. For this purpose, messenger RNA is extracted. From healthy and diseased person. The isolated messenger RNA is converted into complementary DNA. This makes the molecules more stable. The complementary DNA is labeled with one color of fluorescent type. So a microarray chip contains thousands of microscopic spots. Containing millions of copies of specific DNA probe. The labeled complementary DNA samples are mixed and allow to hybrid. So in this way, the microarray detects the disease because mutant alleles or a physically cause the expression of specific genes to be unregulated or downregulated compared to healthy cells is different. It may visualized as a distinct red or green color. So the unhybridized can be washed away. So the gene was expressed more in the control healthy sample. The chip is washed to remove any complementary DNA which did not hybridize. So the microarray detects the disease because mutant alleles or a disease state typically cause the expression of specific genes. So I would like to say it also helps the gene which was not expressed in either. It helps to detect the genes not not expressed. This expression is visualized as red and green pattern on the chips. So in this way, it helps to diagnose, the diseased because mutant alleles or a disease state typically cause the expressions of specific genes. So it helps to find out that. So it was about question part C. Now moving towards the next part D. The nucleotide sequence of alleles that cause genetic disease, such as the various forms of porphyria, are stored in a database. State one benefit of having database of nucleotide sequence for alleles that cause more diseases. So the benefit of genetic sequence database, having a database of nucleotide sequence is that it allows for accurate and rapid diagnosis of specific diagnosis. So clinics are usually compare the DNA sequence from a patient with an undiagnosed condition against the database of non-disease causing mutations. So a match provides a definitive fast diagnosis. So it allows scientists to confirm pathogenicity if a mutant is rare, seeing its documents in a data alongside clinical information from other patterns globally. Scientists, so in this way, the database facilitates research into the molecular cause of the disease by comparing the effect of different mutations or symptoms. So there are many benefits for this, but we have to write only one. The most significant uh, benefit of having a database of nucleotide sequence cause rare disease is that it it allows for accurate and rapid diagnose. It also facilitates research into the molecular cause of the disease. Okay, well, the next part is congenital erythropoietic porphyria is caused by a substitution mutation in a gene coding for an enzyme. The mutant CEP allele codes for an enzyme with little or no function. Scientists use gene editing in the laboratory to correct the nucleotide sequence of the mutant allele in cultured human cells. The scientists introduced three molecules to cells, an enzyme called Cas9 that causes breaks in DNA strands guide RNA attached to Cas9 that is complementary to the mutant CEP allele. In short, a short length of DNA known as template DNA but can replace the section of the allele where the substitution mutation has occurred. The repair mechanism within the cell allowed the template DNA to be inserted so that the mutation is corrected. So figure 3.2 shows part of the nucleotide sequence in the mutated CEP allele before and after the gene editing procedure. So you can see that in the picture, the sequence before and after the gene editing. Okay, you can see the sequence before TAT, GG, CT, CG. You know, the T, thymine, A, adenine, G, guanine, C, cytosine. So these are the nitrogen bases. So the sequence of nucleotides, and in the after the gene editing, you can see T is replaced by C. So in the future, these gene editing procedures may be used in a person with CAP to prevent the accumulation of porphyrins in the body. Suggest and explain how this gene editing procedure will prevent the accumulation of porphyrins in a person with CEP without damaging their genes. So I would go with the answer of this question. So in this, you can say the gene editing procedure prevents the accumulation of porphyrins in this person. So this this is used to correct the underlying genetic defect, restoring the proper function of the enzyme. So this process is based on the CRISPR-Cas9 system and the cell's natural DNA repair mechanism. So first of all, uh, how the mechanism of precise gene correction takes place. So the first point in this targeting the mutant allele. Mutant allele, as per you can see, the T has been replaced by C. So it means this was the mutant T was the mutant allele. So guide RNA is specifically designed to be complementary to the nucleotide sequence of the mutant CEP allele, directing the complex to the exact location of the substitution mutation. So the guide, first of all, the guide RNA is specifically designed to be complementary to the nucleotide sequence of the mutant allele. So it directs the complex at the exact location of the substitution mutation. So the Cas9 enzyme is guided by the gRNA to the target site and acts as a molecular scissor, creating a double strand break in the DNA at the near mutation site on the. So then they repair and uh, correct the cell. Natural DNA repair mechanism is activated, and short length of DNA which contains the correct non-muted nucleotide sequence is introduced and used as a template to repair the break. So this mechanism, known as the homology direct repair, ensures the correct template sequence is inserted, thereby correcting the substitution mutation. So in this way, I would like to write this point, and then the second point, which I'm going, prevention of porphyrin accumulation. So they help by correcting the nucleotide sequence. The gene is now able to code for a functional enzyme of one with the little or no function. So the functional enzyme can now catalyze the specific steps. So in this way, it helps that the precursors will be converted into the next product to be pathway, preventing their accumulation in the cell body. So first of all, the detection of mechanism is used to the correction of precise gene. For which guide RNA is specifically designed. Designed to find out the exact location. And then you can say it in this mechanism, it shows the correct template sequence is inserted. Thereby correcting the substitution mutation. Now the second point is the prevention of porphyrin accumulation. So how it can take place. So the porphyrin precursors will be converted into the next product. Prevention of porphyrin accumulation. It can be done by this means that the porphyrin precursors will be converted into the next product in the pathway. To avoid their accumulation. And the third point can be there is a method uh, avoiding not written properly. Let me erase it. Avoiding damage to other genes. This procedure avoids the damaging of the other genes because it has highly specificity, because the guide RNA has a highly specific that is only complementary to the DNA sequence of the target genes. So this high specificity ensures that the Cas enzyme is only guided to makes a cut at the single intended location in the genome, leaving all other genes undamaged. As gRNA is highly specific to ensure that the Cas9 enzyme is only guided to and make cut at the single location in the genome, leaving all other genes undamaged. So it was all about question number 3. Please like, share and subscribe my channel. Watch more videos on the papers. Thanks for watching.